I don't want you guys to be confused. Okay, two different models. Okay, but you have to look at where these different organelles are protruding from, in order, or coming off of, in order for you to understand what's what. Okay, so here you have what's this structure here?
The nucleus.
The nucleus, the whole thing.
The nucleus.
The whole thing is a nucleus. Okay, and then we have what? What is this?
The nuclear.
That has the pores in it.
The membrane.
The nuclear membrane, right? Or what's that? No, the nuclear envelope, right? One of the two things. Continuous with that is what? So what continue? When I say continuous, meaning it comes off of, but it's still kind of attached to
The rough
rough
ER.
ER, right? The rough endoplasmic reticulum. We know it's a rough ER because of what?
The ribosomes.
Ribosomes. What are those ribosomes producing?
Protein.
Proteins, right? Cytoprotein synthesis. What type of proteins are these? Is the rough ER producing? Proteins that are going to be what?
Used within the cell.
They're going to be either exocytosed or taken out of the cell, okay, transported out of the cell, okay, or they're going to be integrated into the plasma membrane or any one of these membranes on these organelles, right? Whatever mitochondria or any other, or the lysosomes. In the lysosomes, these integral proteins would be hydrogen pumps that are going to be pumping hydrogen into the lysosome to increase the acidity of the lysosomes. Okay, so either case, these ribosomes on the rough ER are making proteins that are going to be either exported out of the cell or integrated into one of the membranes. More specifically, more commonly, the plasma membrane, so that it's easier for you guys to understand. But don't forget that it's also going to be, there could also be proteins that are integrated into the membranes of, say, the mitochondria, because you have things going on in the mitochondria as well. Okay, you have import and export out of the interiors and exteriors of that organelle as well. But when we now look at this guy is what? A smooth ER. How do I know? How do I distinguish it from this guy, which is what?
That's the Golgi.
This is the Golgi. Okay. This guy is continuous, right? Continuous with this rough ER. Okay.
They're attached.
Okay. Attached, right? So that's how you know.
So
Versus
the
this
one on
guy.
the bottom down there, is that the smooth ER on that one?
No.
Or no? That's just something else.
It's still rough because it has
okay.
what?
Oh, the little dots on the outside. Okay.
The ribosomes.
Okay. This guy right here that doesn't have any.
Okay.
He's the smooth.
This would be
The
a smooth.
smooth
So it
ER.
doesn't necessarily has to be attached to.
Well, it is. You have to understand, okay, if you look at the picture in the book, okay, you have to understand that these models are two-dimensional, or they're trying to be three-dimensional, but they're really giving you a two-dimensional picture, okay? They might not be continuous when you're looking at them here, but they could be continuous if you're looking at it from another angle, okay?
So we just need to know that because it doesn't have the little dots.
It's a smooth, okay? Now, again, you would know that it's the Golgi because it's not continuous with any one of these structures, and you have these little sacs, okay? Where what's going on inside these sacs? On this end of the Golgi apparatus, okay, this end is called what? The cis or the trans? The cis. The cis face of the Golgi apparatus receives the vesicles from the rough ER, okay? Whereas the trans face will release the vesicles that have proteins that have been modified already, or lipids that have been modified already, okay? And so it goes one of three places. One place would be what? That's one. It could be the plasma membrane or any one of these membranes, okay? Or it could do exocytose, right? So it's going to have, imagine a vesicle with a bunch of different, proteins inside or enzymes inside, and it's going to fuse on into the plasma membrane and then release them or export them out of the cell. Or if it's not being exported and it's not being formed into a lysosome, what is it doing with those proteins? It's integrating them into the plasma membrane or these other membranes here. Okay, so you have three things: either export of the proteins, integration into the membrane, or formation of a lysosome for these.
So those little orange dots next to them, those are the lysosomes.
These guys, yes, right here.
And the ones next to the Golgi, like
That's
alongside the little ones?
those would be. Imagine
Still
these
part
are
of
those
the-
vesicles, and it's gonna you don't know that where it's gonna go, but know that it's gonna go into forming one of these, exporting it out, or integrating into the plasma membrane. Make sense? Yeah. Okay.
Can I take a picture really quick up close? Sorry, I'm in your way. Go.
Okay, go ahead. No, go.